In our previous video looking at Krebs cycle, we had just finished the second dehydrogenation reaction and production of an NADH molecule, leaving us with this rather strange looking intermediate here. So we can see that the decarboxylation of this CO2 here means that there is no longer a CO2 on this side of the molecule right here, and we can see that there is only a single hydrogen remaining on this carbon here with a double bond previously in its place. Now, this intermediate exists for a very short period of time because our coenzyme A is going to come into play here. So as a consequence of what happens here, the sulfur can actually form a bond with this carbon here, causing this double bond to reform and attach itself to the hydrogen here. And when we redraw this structure here, we end up with the molecule succinic acid bonded to coenzyme A, which we can henceforth refer to as succinyl-CoA. Now, once again, coenzyme A needs to be a recyclable molecule, so its binding here is only temporary, specifically in order to create a large enough positive charge to attract another phosphate ion. So here we are preparing for yet another example of substrate-level phosphorylation, as we saw twice in glycolysis here. So because of the fact that this carbon is bonded to the more electronegative sulfur and the highly more electronegative oxygen, it creates a strong enough positive charge in which the coenzyme A can actually be broken off when a phosphate physically makes a new covalent bond with this carbon here, breaking off the sulfur group and therefore releasing coenzyme A in order for it to perform its original functions. Now, as with many cases involving substrate-level phosphorylation, if we take a look at this phosphate here, we see that there are no examples of positive poles anywhere to be found in order to stabilize this negative charge, and not only is it close to a strongly electronegative oxygen, but we also have a double bond that has, because a double bond is two bonds and each bond is two electrons, that means that there are four electrons that strengthen the repulsive force here, making this phosphate very unstable and allowing it to be removed by yet another adenosine diphosphate molecule here. And again, when this is broken off, this phosphate will join with adenosine diphosphate in order to make ATP, in yet another example of substrate-level phosphorylation. Now, all that remains of Krebs cycle is actually to get back to the starting point of the cycle at which acetyl coenzyme A joined the reaction. Now, the problem is with this particular molecule here is that the center is almost completely nonpolar, which is why it requires a specific electron carrier molecule called flavin adenine dinucleotide, and for those that can't sound out the word flavin, here it is. So flavin adenine dinucleotide is an electron carrier molecule similar in function to nicotinamide adenine dinucleotide, only it specializes in taking hydrogens, in this case two hydrogens, and an additional pair of electrons from more nonpolar areas, such as what we see here. And what is ultimately going to happen here is that one of the hydrogens is going to be removed and its bond will be relocated over here to form a double bond, therefore breaking one of the additional hydrogens and its two electrons off, and having that join with flavin adenine dinucleotide, FAD, in order to produce FADH2, which again is functionally analogous to NADH, in that its primary function is a carrier of electrons and a method of transferring potential energy from Krebs cycle to the next part of cellular respiration, the electron transport chain. And after this reaction is completed, we end up with another compound that we call fumarate, to which a very simple addition of a water molecule is done. So here, because of the fact that a double bond is essentially 4 electrons, that means that there's a large amount of repulsive force in here, so the double bond can actually attract to the positive pole on the hydrogen of water, allowing the hydrogen to add to itself here. Now, as a consequence of this, the OH side of water can then add onto this side here, giving us our penultimate compound in Krebs cycle, malic acid or malate. And this is where our final dehydrogenation reaction happens here, but this dehydrogenation is a little bit different from the ones that we saw previously because there is no removal of carbon dioxide. There is no decarboxylation that occurs here. So in this particular reaction, the enzyme in this reaction is going to pull this particular hydrogen off, turning this bond into a lone pair. And when this oxygen has 3 lone pairs, that lone pair is going to come down and form a double bond like this, causing this carbon to have 5 bonds. Therefore, this bond with its 2 electrons can break off, join with hydrogen, and then both of the electrons in the pair and H plus can join with NAD plus in order to give us our final molecule of NADH produced in Krebs cycle. And if we look at the molecule that we end up with here and compare it to the molecule that we started with at the very beginning, we see that this is the same molecule, and therefore that we have made it all the way back to oxalic acid or oxaloacetate, which is the starting point of Krebs cycle. So to conclude this video, if we do a whirlwind tour of Krebs cycle, just to summarize all of the important points, we start with the addition of acetyl coenzyme A here in order to form citric acid, which is promptly isomerized by taking this OH group and moving it over here. And then this is where the first of our decarboxylation reactions occur, where this carboxyl group is broken off to make CO2, and the enzyme that removes this hydrogen here triggers this hydrogen to come off with its electrons in order to form the first of our NADH molecules. And then in the next phase, that process happens once again with our decarboxylation happening here. So this is our second carbon dioxide that is lost, resulting in a rearrangement of bonds, breaking this particular hydrogen off with its electrons for our second NADH molecule. And this allows coenzyme A, which we can write in here, this allows coenzyme A to come in and make a bond here, and this is our prime opportunity for our substrate-level phosphorylation to take place. So this phosphate is going to come and add itself here, breaking off coenzyme A, and then allowing ADP in order to come in here, phosphorylate itself by taking this reactive inorganic phosphate like this and becoming ATP, leading us to the closing phase where FAD takes 2 of these hydrogens and 1 pair of the electrons here, forming fumarate, which we can then convert into malate by adding the OH onto this carbon and the H from water onto this carbon, leading to the final NAD plus reaction here, in which this OH becomes a double bonded oxygen. This electron pair breaks off, taking this hydrogen to join NAD+, giving us our third NADH molecule. Now, if we do a complete tally, we can see per one cycle of Krebs cycle, we generate 1, 2, 3 molecules of NADH and 1 molecule of the closely related flavin-adenine dinucleotide FADH2 with 1 molecule of ATP produced through substrate-level phosphorylation. But we remember that per glucose molecule means that 2 cycles of Krebs cycles occur because there were 2 pyruvic acid molecules that were produced at the end of glycolysis, so we can multiply each of these steps by 2 in order to give us our total products. And in the final series in the cellular respiration unit, we will look at what all the NADH and FADH2 is used for.